Connaissance.
Recherche.
Sciences.
Des sciences mathématiques.
Théorie.
Physique.
Philosophie.
Conférence. Archéologie.
Environnement. Nature.
Mesdames et messieurs, c'est un plaisir d'ouvrir cet après-midi la session ici au Collège de France et je remercie de ça les organisateurs. Before starting my talk, I would like to argue about the term non-conventional. So non-conventional so actually the term conventional is associated usually to what is a common belief. It follows tradition and gives a sort of feeling of protection against unknown. Instead, non-conventional has very often a negative meaning and is also associated with a negative perception. I do not really agree on this, especially in science, where non-conventional things, for me, have a positive meaning. They are synonym of novelty, of innovation, and also a way of looking at simple solutions and simple answers, as opposed to maybe complicated argumentation to follow convention. I'm going to talk about the lipid antigen recognition by the immune system. And lipid antigens are considered non-conventional antigens simply because they were discovered much longer after the peptide antigens. Now, the fact that they have been discovered as second should not affect their importance. It is, if you wish, like in a sport contest, not always the second is less excellent than the first one. So, Docet Omnia is the motto of the Collège de France. If I may, I would like to change it today in Omnia Docent, which is quite appropriate with the title of the conference, of today's conference. And this will focus in a way the attention to those non-conventional observations that might induce changes in the current scientific dogmas. So T cells recognize lipid antigens, and they do it with the T cell receptor, which sees the complex of lipid associated with CD1 molecules. The fact that this kind of immune response was not known before was because the function of the CD1 molecule has been characterized only relatively recently, so something like about 10 years ago. CD1 in humans are 5 different molecules from A to E. They are glycoproteins which resemble class 1 MHC molecules. Their extracellular domains are constituted by alpha 1, alpha 2, and alpha 3 domains, and they associate with beta 2 microglobulin in a non-covalent way. In humans, they are expressed by the antigen-presenting cells, so dendritic, macrophages, Langerhans cells, as well as by thymocytes and B cells. Their function is to bind lipids, and this is an example of a sulfatide inserted into the CD1A molecule. And if we have the exception of CD1E, which stays only intracellular and never traffic to the cell surface, they all present lipid to T cells. This is the crystal structure of CD1-D complex with a prototype antigen, alpha-galactosyl ceramide. This is the site where the complex is inserted into the plasma membrane, and this is the site where it takes contact with the T-cell receptor. Now, already from this first projection, one can see that the lipid antigen protrudes very, very little out of the surface of the old CD1 molecule. Sorry. Because it is actually all, the lipid is all contained into the antigen binding pocket that is created between alpha-1 and alpha-2 helices. And this pocket is very hydrophobic and contains, therefore, the lipid hydrophobic tails. If I zoom into the antigen binding pocket of these molecules, now it's clear how the antigen, the lipid antigen, is associated by these hydrogen bondings that keep the lipid in exact position, which is very important for the stimulation of the T cells. The importance of the lipid T cell response is, in my opinion, illustrated here. Differently from MHC molecules, CD1 molecules are not polymorphic. And therefore, one can expect a T-cell response at a populational level. Second important feature is that lipids derive from a complex enzymatic synthesis pathway, and therefore they are not easily modified by mutagenesis, which can be induced by selective pressures. Third important point, and that we and others have found high frequency of lipid-specific T cells in the peripheral blood of normal individuals. So we are very much interested in the use of immunogenic lipids as vaccines. And I would like to give you an example of bacterial lipids as vaccines. This is a new antigen purified from Mycobacterium tuberculosis. It is D-acyl trialose sulfate. And the important characteristic of this lipid is that it is expressed only by virulent mycobacterial strains. What is relevant for a vaccine is to evaluate the extent of the response during infection with mycobacteria. Here we measure the reactivity of freshly isolated peripheral blood mononuclear cells in response to the antigen sulfoglycolipid. PPD negative as well as PPD positive individuals have been taken, and patients with pulmonary active tuberculosis. Now, as you can see now here, the response measured as interferon gamma of the T cells in the peripheral blood demonstrates that there is a correlation with a previous contact with tuberculosis, with Mycobacterium tuberculosis. What is important next for a protective immune response is that there is a killing of the intracellular bacteria. So for this reason, we have made T-cell clones out of those peripheral T cells and characterized their reactivity against sulfoglycolipid. All the clones, or the majority of the clones that we found, turned out to be CD1b restricted. And so in the assay that I'm going to show you, we measured killing of not only of the infected antigen-presenting cell, but also of the microbacteria, intracellular microbacteria. And these are the results. This is the antimicrobacterial activity of these T cells evaluated as surviving colony-forming units of the bacteria. And these T cells are able to kill the intracellular microbacteria after 72 hours of interaction. And there is more killing depending on the amount of T cells that we add to the assay. So immediately after those results, we decided to go into a sort of translation of our information in a vaccination setting. And we used guinea pigs immunized with a mixture of sulfoglycolipid and PIM6. PIM6 is a phosphatidyl inositol as a monocyte and is another lipid antigen present in Mycobacterium tuberculosis. Animals were challenged with virulent mycobacteria, and the survival of our experimentally vaccinated animals with the two lipids is better than the one we observed with the current Mycobacterium tuberculosis vaccine, the BCG. The reason why we use guinea pigs is that they do express CD1b, whereas mice and rats do not express this molecule. And for this reason, instead of repeating experiments which last 300 days, we have made transgenic mice for the human CD1b molecule, and we are going to repeat all these vaccination experiments in our transgenic mice. Now, the second part of my talk deals with the apparent paradox of how the large lipid antigen repertoire can be displaced and recognized by the immune system, because CD1 molecules are not polymorphic. And nevertheless, they do bind a large variety of antigenic lipids. These are examples of antigenic lipids. Lipids are amphipathic molecules, so they have an hydrophobic as well as an hydrophilic part. And there are several examples of these antigenic lipids in which there is one hydrophobic or two hydrophobic chains. They can have different lengths. For example, in the case of sulfatide, the ceramide is quite short, whereas in the case of glucose monomycolate, they can have up to 80 carbon atoms, so quite long. They can have also different hydrophilic heads, like a very short, small carboxylic group on the mycolic acid, until a pentasaccharide that is on top of ganglioside GM1. So actually, how do CD1 present lipid antigens with such different structure? The first strategy was to have different CD1 molecules with different antigen binding pockets. For example, CD1D or CD1A have these two pockets that accommodate the lipid antigens. A very intricate pocket is instead the one of CD1, human CD1B. It has A prime, a C prime, and F prime pockets, and a tunnel connecting A prime and F prime pockets. The structure of the human CD1B has been resolved by crystal analysis, and this is the native CD1B antigen-presenting group without exogenally added antigens. The pockets appear to be all filled by some molecule. Actually, this is the molecule occupying the A prime and the C prime pocket in the native CD1B, which I forgot to say before has been expressed in mammalian cells, is occupied by phosphatidylcholine. This has been identified as phosphatidylcholine. And the F prime and the tunnels are occupied by a known spacer molecule which is 40 to 44 carbon atoms in length. So we believe that these molecules contained into the pocket of the CD1B are important for maintaining the structure of this complicated groove. When we load the soluble recombinant CD1B molecules with the sulfoglycolipid antigen of Mycobacterium tuberculosis, now the phosphatidylcholine is replaced by the AC2SGL antigen, and at the same time, the spacer shifts inside the tunnel and slides in a way to allow a better fitting of the antigen. At the same time, the spacer molecule, which is pointing upwards in direction of the interaction with the T-cell receptor, now here is completely inside the groove and does not protrude anymore. The structural changes that are associated with antigen binding are not just the one of the spacer, but we found another two important structural changes in the CD1B molecule. This is a view from the top of the groove. In blue, the loop indicates the native CD1B molecule, and in white, instead, the one loaded with the sulfoglycolipid antigen. These two amino acids, the glutamic acid 80 and the tyrosine 151, become in the antigen-loaded conformation closed, indicating that actually the F' pocket is now closed. And another important change is that the asparagine 152 from an horizontal position in the native CD1B now flips vertical in the direction of the T-cell receptor binding site. So this important, how important are these structural changes in terms of function? In order to understand this point, we have generated antigen-presenting molecule expressing CD1B mutated in those three residues that we had identified by crystal structure. And we have used these mutants for presentation of sulfoglycolipid to specific T cells. These are the results. This is the normal response at increasing doses of sulfoglycolipid antigen presented by wild-type CD1B molecules. And instead, the response is almost completely abrogated when we mutate E into alanine and tyrosine into alanine, suggesting that these two residues, which are really closing the pocket, the F' pocket, are important for the recognition of the T cells. The mutation of Q152 into alanine is instead favoring the presentation, suggesting in this case that this residue, which flips horizontal, can make steric hindrance. Now removing it to a smaller amino acid like alanine makes the interaction with the T-cell receptor and the stimulation of the T cells even better. So, in summary, I would like to propose the following model. When we have native CD1B molecules, the antigen groove is occupied by endogenous phosphorylcholine and a spacer molecule. When we load the CD1B with antigen, now the phosphatidylcholine is exchanged and is substituted by the antigen, and the spacer slides inside to favor the accommodation of the antigen. And now the complex becomes stimulatory for T cells. The conclusion is that all these structural changes that I've shown you allow a presentation of structurally different lipid antigens. And most importantly, they allow the immune system to discriminate between antigen-loaded and antigen-unloaded CD1b molecules. This has important consequence and meaning because these results would suggest that plasticity in an antigen-presenting molecule can be an alternative strategy to polymorphism. I would like to thank at the end my collaborators who participated in the work, and this is the group of the present and past group of Gennaro De Libero in Basel, the group of Germain Pouzot and Martin Gilleron in Toulouse, who contributed to the synthesis and characterization of the glycolipid of mycobacteria, Luis Garcia and Lionel Mouret, who made the crystal structure of the CD1b molecule, Kees Vers-Louis and Albert Heck made the mass spectrometry data, which I did not present, but were important for the identification of the spacer molecule inside the CD1b. And Stefan Stenger and Anne Rokins participated to the infection experiment with virulent mycobacteria. And I thank you for your attention this afternoon.